jsem dělal, to nešlo. Teď to jde.
Tak můžete povídat. Můžete povídat.
First group of the neurotransmitters first system is localized in the hemisphere and in the brainstem is cholinergic system, which is divided to 6 neuronal groups, and these cholinergic nuclei produce acetylcholine. Acetylcholine exhibits generally excitatory influence to the other neurons. So acetylcholine is distributed from cell bodies which produced of acetylcholine to their target areas, and target areas are or in the cortex or in the thalamus. So this picture shows schematically position of main cholinergic groups. And you see that first group of cholinergic neurons is localized in the septum verum. The same is for the cholinergic group 2, which is also in the septum verum. In the cholinergic group 3 is situated in the diagonal area, which is a small area in the base of the hemisphere, and largest cholinergic group of the hemisphere is cholinergic 4, which is localized in the basal nucleus of the mynad. Basal nucleus of the mynad is situated in the sublenticular area of the hemisphere, so below the lentiform nucleus, putamen and globus pallidus, between the surface of the hemisphere and these two strata nuclei. And arrows also indicates the distribution of the synthesized acetylcholine. So from group 4 is acetylcholine distributed to the whole neocortex, to the temporal neocortex and to the amygdala. Acetylcholine, which is produced in the groups 1 and 2, is distributed to the hippocampal formation, and cholinergic group 3, which is localized in the diagonal area, sends axons and distribute acetylcholine to the olfactory areas of the brain and to the olfactory bulb. So these 4 cholinergic groups supply acetylcholine to the whole neocortex, to the neocortex of the whole hemisphere, and in addition to olfactory bulb and olfactory cortex and to the amygdala. Last 2 groups are designated as a cholinergic 5 and cholinergic 6. These cholinergic neurons are situated in the brainstem, in the reticular formation of the brainstem, in transition between the pontine reticular formation and midbrain reticular formation. And group 5 is localized in the pedunculopontine nucleus, and group 6 is localized in the laterodorsal nucleus of the brainstem of the reticular formation. These 2 cholinergic nuclei project to the thalamus and supply thalamus with acetylcholine. So production of the and distribution and transportation of acetylcholine from group 5 and 6 to the thalamus results in excitation of the activation of the thalamic neurons, and due to the fact that the thalamic neurons are intensely interconnected with the cerebral cortex, production or distribution of the acetylcholine, in addition to activation of the thalamus, activate also cerebral cortex. So all these systems, in fact, activate the cerebral cortex. Brainstem groups activate also thalamus. This picture shows cores of the cholinergic neurons, which originates in group 4. It is minor nucleus, basal nucleus of the minors. It is designated as a group 4. And this basal nucleus of minors is situated below the lentiform nucleus, which is formed by the putamen and globus pallidus, and basally to these two nuclei is anterior commissure, and basal nucleus is just under the anterior commissure, between the commissure and lentiform nucleus and surface of the hemisphere. An arrows indicates general distribution of the cholinergic fibers. You see that cholinergic fibers emerge the lateral margin of the basal nucleus, and part of this bundle terminate in the amygdala. Rest of the basally oriented fibers terminate in the temporal neocortex, and ascending limb of these projection terminate in the insular cortex, in the cortex of the parietal and frontal lobe, and in the medial aspect of the hemisphere, where is singular gyrus and superior frontal and other gyri of the medial aspect of the hemisphere. In the deep to singular gyrus, cholinergic fibers form part of the bundle of associative fibers, which is named cingulum, and cingulum passes antero-posteriorly along the whole length of the singular gyrus and supply as cholinergic fibers medial aspect of the hemisphere. So generally, it's possible to say that the cholinergic group 4, which is placed in the basal nucleus of the minor, supplies neocortex and amygdala. Schema, generally mammalian schema of the cholinergic system, and it is demonstrated in the brain of schematic picture of the brain of the rat, laboratory rat. The individual groups, this is septum verum, containing cholinergic group 1, 2, and you see that these two groups project to the hippocampal formation. Hippocampal formation is cortex, but it is 3-layered primitive cortex, developmentally very old, and generally cortices doesn't exhibit its own source of the acetylcholine. So acetylcholine must be transported to cortices via cholinergic projections, via this system. So groups 1 and 2 project and influences activity of the hippocampus. In contrast to the group 4, it was demonstrated in previous picture, supplies the whole neocortex, and group 3 supplies the olfactory bulb and olfactory cortices. In addition to cholinergic projection of group 1, 2, 3, 4, we must also mention that striatum contains its own cholinergic system. These are large neurons with short axons. So these neurons, cholinergic neurons of the striatum, are local interneurons and supply of the striatum with very high density of cholinergic terminals and supply the striatum high concentration of the acetylcholine. Let us return back to the brainstem. There are two groups, 5 and 6, and these two groups supply the thalamus. Result of influence of the acetylcholine in the thalamus is activation of the thalamic neurons, and of course also thalamocortical neurons, which project to the cortex. So resulting is also activating of the whole cortex or specific areas of the neocortex, which are supplied by thalamic nuclei. So thalamocortical system is activated from two cholinergic systems of the brainstem. So this table shows localization of the cholinergic neurons, and right half of this table indicates the which structures are influenced from respective groups of the cholinergic neurons. So, cholinergic group 1, 2, which is in the septum verum, project to the hippocampus, to the cortex of the hippocampus. Group 3, which is situated in the diagonal area, the basal part of the hemisphere. It is in human brain, it is small area, influence the olfactory bulb. And group 4, which is placed in the basal nucleus of the minor, it supplies whole neocortex and amygdala. And brainstem groups 5 and 6, which are localized in the reticular formation, so laterodorsal nucleus and pedunculopontine nucleus, influence or activate the thalamus. So these fibers emerging of cholinergic neurons of group 5 and 6 terminate in the thalamus and influence activity of the thalamic neurons and activate generally thalamus. Due to the fact that thalamus contains many thalamocortical neurons, resulting is also activation of the cerebral cortex. So cerebral cortex is activated by two sources. First one is the basal nucleus, is group 4, and second source of the activation is via thalamus from brainstem cholinergic neurons. Generally function of the acetylcholine. Acetylcholine augments the excitability of cortical neurons, improve the circulation. Acetylcholine is responsible for slight dilatation of the cortical vessels, and due to improved circulation supports the memory mechanism. So decrease of cholinergic neurons of the brain and the decrease of, which is associated with the decrease of production of the acetylcholine, leads to the problems of memory mechanism and problems of circulation, blood circulation in the cerebral cortex. So degenerative diseases affecting cholinergic groups result in these memory problems and vascular supply of the brain. Another wide distributed and very extensive system is serotoninergic system. Serotoninergic system is localized in the Raphael nuclei of the reticular formation, and at approximately half of these neurons produce the serotonin, and serotonin is distributed to the thalamus, hypothalamus, to the basal ganglia, and to the whole neocortex. And in addition to cortical structures, serotonin influences the amygdala, is number 2, and number 3 is hippocampal formation. Lower serotoninergic neurons of the reticular formation supply the cerebellum with serotonin, and also there is a group of caudally localized neurons of the Raphael nuclei of the reticular formation, which project to the spinal cord. So this is very extensive projections influencing many very important systems of the brain. So if we summarize it, so serotonergic system is localized in the reticular formation, in the Raphael nuclei of the reticular formation. Okay. Generally, this system is divided to ascending and descending component. Ascending system supply all cortical formation, is the neocortex, the cortex of the limbic system, basal ganglia, thalamus, hypothalamus, and in addition to telencephalic structures, serotonin is distributed also to the brainstem, to the cerebellum, and to the spinal cord. And if activity in the descending system of the serotonin system, which project to the spinal cord, result in analgesia. So pain is perceived in a reduction form. Decrease of synthesis of serotonin leads to depression, irritability, sleep disorders, and insomnia. Increase of synthesis result in tremor, trembling, increase of blood pressure and heart rate, confusion, and unconscious. Important is influence of the sunlighting. Sunlighting increases production, and this is it will be demonstrated in the situation of inhabitants living in the northern Europe and northern Canada, northern Russia, where it's long winter, dark winter, the sun lighting is reduced to several hours only, and people living in these areas are affected very frequently by depression, increased irritability, and eventually anxiety. Serotonergic system is part of the monoaminergic system, and second neurotransmitter belonging to this monoaminergic system is noradrenergic system, while serotoninergic were produced in the Raphael nuclei of the reticular formation. Noradrenergic system is produced in groups which are designated as A, from A1 to A7, and A7 is largest group of neurons, and this is named locus coeruleus. Position of the locus coeruleus is evident in the fourth ventricle, where is possible to find small spot which is bluish in color, and this color is caused by the accumulation of the neurons which contains pigment and dark pigment, and this pigment is visible under the floor of the fourth ventricle, and macroscopically it is designated as a locus coeruleus. Noradrenergic groups project to the spinal cord, to the sensory nuclei of the cranial nerves, mainly to the trigeminal nucleus, after that to the cerebellum, hypothalamus, and to the thalamus. But in the thalamus, receive noradrenergic fibers, sensory thalamic nuclei, ventralis posterolaterales, ventralis posteromedialis, and lateral geniculate body. And cortical distribution is to the whole neocortex and to the hippocampal formation, but noradrenaline is not distributed in the striatum. So noradrenergic system doesn't project to the striatum. So let us summarize the noradrenergic system. Main productor is group of neurons designated as A6, which is, for this group is used term locus coeruleus, and fibers emerging of noradrenergic cells are terminated in the spinal cord, in the brainstem, in the cranial nerve sensitive nuclei. Main nucleus is trigeminal sensitive nucleus. Noradrenergic system influence cerebellum, specifically the Purkinje cells. Part of the noradrenergic projection terminates in the hypothalamus, and in the thalamus, in the VPL, VPM, and geniculate bodies. And finally, final destination within the hemisphere is neocortex and hippocampal formation, and the noradrenergic system is part of the ascending activation system of the reticular formation. So function of noradrenergic system is regulation of transmission of sensory signals, pain. Let us remember that trigeminal nucleus receives many fibers. The same is true for the posterior horns of the spinal cord. Another function is in the regulation of circulation, and this system is important to feel energized. So if production of the noradrenaline is optimal or relatively higher, people feel to be energized, are very active. Decrease in production of the noradrenaline leads to feeling of exhaustion. People are tired and reported that they are without energy. So, and another system is dopaminergic system of the brain. The dopaminergic system is very important due to the fact that it is associated with the Parkinsonian disease, and dopaminergic cells, which synthesize and produce dopamine, are located in substantia nigra, pars compacta. It is pigmented part of the substantia nigra, and this, according to numerical code, nigra compacta is designated as A9. Medially to substantia nigra compacta is ventral tegmental area, which is designated as a group A10. Fibers emerging of these two structures project and terminate in the striatum, in the pallidum, in the neocortex, and decrease in production results in Parkinsonian syndrome or Parkinsonism. Patients which are the problem is the production of the dopamine. If level of the dopamine is decreased, dopamine may be replaced by precursors of the dopamine. It is L-dopa, and L-dopa is converted in the brain tissue to the dopamine. Another large system is glutamatergic system. Glutamate is excitatory neurotransmitter. Glutamate is distributed in majority of ascending and descending pathways. For example, all descending cortical pathways like corticostriatal, corticothalamic, corticospinal, cortical projection terminating in the brainstem in the reticular formation, all contains glutamate, which is produced in cells of origin of this system. Another source of the glutamate are descending brainstem pathways like vestibulospinal, like reticulospinal, rubrospinal, and also glutamate is distributed in the afferent cerebellar pathways. So afferent cerebellar pathways originate in the cerebral nuclei. Largest of them is dentate nucleus, and projection of the dentate nucleus terminating in the thalamus. So dentate-thalamic projection is glutamatergic projection. Similarly, commissural pathways, so all fibers in the corpus callosum and in associative cortical pathways, all are glutamatergic. Another large system is GABAergic system. In contrast to glutamatergic, which is excitatory neurotransmitter, we may say that it is prevailing neurotransmitter in the whole brain. GABAergic system is inhibitory system, and GABA is inhibitory neurotransmitter. GABA is produced in local interneurons, which are located in the neocortex, in the hippocampus, in the thalamic nuclei, in the cerebellar cortex. In the cerebellar cortex are GABAergic elements, Purkinje cells, of course, but also neurons located in the layer 1 of the cerebellum. Basket cells and stellate cells are also inhibitory. So, in addition to local interneurons, GABA is produced also in the striatal and transported in striatal efferent projections. So, efferent neurons of the striatum, which are medium-sized spiny neurons, these cells produce the GABA, and GABA is conveyed to the targets of this projection, to the globus pallidus and to the nigra reticulata. Similarly, globus pallidus is also consists of neurons which produced GABA or GABAergic, GABAergic, and efferent projections of the globus pallidus. For example, globus pallidus externus project to the subthalamic nucleus. These projections is inhibitory, inhibits subthalamic cells. Concerning the internal segment of the pallidum, pallidum internum, projection, this projection of the pallidum internum is terminating in the thalamus, also are inhibit thalamic neurons and consecutively thalamocortical projections. Thalamus, in addition to local GABA interneurons, is exhibit another supplier of the GABA, and it is not reticular nucleus of the thalamus, which is most lateral flattened nucleus, which is between the internal capsule and other thalamic nuclei. So all the neurons of the reticular nucleus of the thalamus are GABAergic, so belong to the GABAergic system. In the last 20 years was discovered that in addition to above mentioned neurotransmitters, there is also gaseous neurotransmitter, which is nitric oxide, which is produced by enzyme, which is named nitric oxide synthesis. Nitric oxide synthesis may be neuronal, which is produced by neuronal NOS. After that, in the endothelial cells, because these cells produce the endothelial NOS, and in a situation where CNS is affected by some pathology, some neurons of the and some glial cells produce also NOS, so nitric oxide, and contains enzyme which is responsible for the production of the NO. Nitric oxide, which is released of the cell body of the neurons, is distributed to distance 200 microns to sphere, 200 microns from the cell, respective cell body, or cell body positive for NOS. And in this area is estimated 2 million of synaptic contacts. So NO may influence quite large amount of the synaptic contacts in the vicinity of the NOS positive cells. Concerning the function, NO increases releasing of neurotransmitter, so you may say that activate some kinds of the cells, and is responsible for the vasodilatation. So endothelial NOS produce large amount of the NO, and NO exhibit the dilatation of the vessels, relaxation of the vascular musculature, and final dilatation of the vessels. In several neurodegenerative diseases, this production of the NO increased, and this increased production may result in damage of neurons. Another topic of our course is vasculature of the CNS. So vessels supplying by arterial blood, the CNS, and on the other side, vessels, veins, which convey the venous blood to the general circulation. So, CNS is supplied by two vascular systems. First one is vertebrobasilar system. And vertebrobasilar system originates in the vertebral artery, which is branch of the subclavian artery, first part of the subclavian artery. After that, vertebral artery enters to the foramina in the vertebral transversal processes, and from C6 to C1 passes through this foramina, and above the atlas enters to the foramen magnum, and right and left vertebral artery join and form the basilar artery, and basilar artery, which is situated in the pons varoli, in the basilaris sulcus of the pons, is divided to two strong arteries, which are named cerebral posterior arteries. So, vertebral artery gives off muscular branches for the deep neck muscles, and in addition to muscular spinal branches, rami spinalis, which enters to the intervertebral foramina and co-supply spinal cord. In addition to these branches, this system gives off arteria spinalis posterior, so posterior spinal artery, which is paired artery, right and left, and anterior spinal artery. After that, artery supplying the oblongata, medullary arteries, and arteries, one pair of arteries supplying the cerebellum. It is anterior cerebellar, it is cerebral artery inferior posterior. So posterior surface of the cerebellum is supplied by this artery. So this picture shows anterior spinal artery, which originates by two branches emerging of the vertebral artery, and these anterior spinal artery supplies it is very long artery. Its continuity may be interrupted and replaced by a small plexus of the arteries. And due to the fact that this artery is relatively thin, its volume is small, spinal cord must be supplied by segmental rami spinalis or radicular arteries, which improves the supply of the spinal cord by the arterial blood. Anterior spinal artery gives off one larger branch, which is named Adamkiewicz artery or Arteria spinalis magna, which emerges of the anterior spinal at a level between the 7 thoracic vertebrae and 2nd lumbar vertebrae. So in this part of the spinal cord, this part of the spinal cord may receive the Adamkiewicz artery, which also helps to supply the lumbar and lower thoracic parts of the spinal cord. In addition to anterior spinal artery, there are two posterior spinal arteries, so arteria spinalis posteriores, and anterior and posterior spinal arteries are interconnected by arcades, arterial arcades, which gives off many smaller branches for supplying of the mainly white matter of the spinal cord. A gray matter of the spinal cord is mainly supplied by of anterior spinal artery, and posterior horn may be co-supplied from the posterior spinal artery. Next part of the brain circulation is basilar artery. After joining of the right and left vertebral artery, constituted the basilar artery, situated, placed in the basilaris sulcus of the pons varolii, and this artery gives off branches for the medulla oblongata, branches for varolian pons varolii, branches, arteries for the labyrinth, which enter porus acousticus internus together with the intermediofacial nerve and vestibulocochlear nerve. And next are arteries for the cerebellum, arteria cerebellaris inferioris, anterioris, and arteria cerebellaris superiores. And last branches are arteria cerebri posterior, and arteria cerebri posterior is largest of these above mentioned and gives off posterior communicating artery. So posterior communicating artery interconnects the basilar artery with the system of internal carotid artery. Schematic picture of the longitudinal system of the spinal cord, so anterior spinal cord artery and two posterior spinal cord artery. But from the picture is evident that this longitudinal system is relatively big, is formed of the thin vessels, and must be circulation in the spinal cord must be improved by the radicular arteries, which are joined with the anterior and posterior spinal cord artery. As we mentioned formerly, basilar artery is divided to posterior cerebral arteries, right and left, and posterior cerebral gives off posterior communicating artery, which joins the posterior cerebral with the internal carotid artery. And so posterior communicating interconnects the two systems, carotid systems and vertebro-basilar system, and this interconnection is completed by anterior communicating artery, which interconnects the anterior right and left anterior cerebral artery. So this connection of the vessels of the base of the brain, which is around the hypophyseal stalk, is named Circle of the Willis. And Circle of the Willis gives off cortical branches, which are three: anterior cerebral, middle cerebral, which is largest, and posterior cerebral. But in addition to these large cortical arteries, there is many smaller, so-called basilar arteries or central arteries. And there are several groups of the anteromedial group of the central arteries, anterolateral group of the central, posterolateral and posteromedial. These central arteries supply deep structures of the brain. It is basal ganglia and thalamus and hypothalamus. So large vessels supply the cortex, smaller branches supply subcortical gray matter in the basal ganglia and in the diencephalon. Circle of Willis represent the communication between vertebral and anterior carotis arteria carotis interna systems. So these two systems, vertebral, vertebrobasilar, and arteria carotis interna are interconnected. Interconnection is formed by anterior and posterior communicating arteries. So these communicating arteries allow blood to flow between both systems. So posterior communicating artery interconnecting vertebral basilar and carotid system, or between the right and left vessels, and this connection between the right and left half of the Willisian circle is established by anterior communicating artery. Circle of Willis exhibit large variability. Only 60% of cases shows normal anatomical arrangement of the vessels, but in other situations there are some problems. So this picture, which is evident in the 10% of cases, shows a very thin anterior cerebral artery, which is in fact supplied by the anterior cerebral from one side, which is divided and follows the course of the classical anterior cerebral, but main source is reduced, is very thin. Another case is in volume, are changes in volume of the posterior communicating artery. In the 10% is very thin posterior communicating on one side, and in the same number of cases is reduction in volume of the both posterior cerebral communicating arteries. And this picture shows another case which indicates very thin one posterior cerebral, while the other side it is in normal size and in normal distribution. So variability is large, and only 60% of cases exhibit classical arrangement of the Willisian circle, which represents a normal situation. Concerning the arteria carotis interna, arteria carotis interna exhibits several portions. So it is cervical portions which terminate at the base of the skull. After that, artery enters to the carotid canal, and this part is named pars petrosa. Following is short part of artery which is played in the cavernous sinus. It is along the sides of the body of the sphenoid bone. It is pars cavernosa. And last portions of the carotis interna is cerebral part. So these are arteria cerebri anterior and media, and arteria ophthalmica, and arteria chorioidea anterior. It is reconstruction, 3D dimensional reconstruction of the Willisian circles, which was obtained by the basic data are from the CT examination. So evident is right and left vertebra, basilarity artery, posterior cerebral artery, right and left, communicating arteries, one, and it is internal carotid artery, which gives of anterior cerebral and middle cerebral, and this is small anterior communicating artery, which is relatively thin. And the most important aspect of the study of the circulation of the brain is distribution of its cortical branches. So this picture, the lower picture shows distribution of the middle cerebral artery. You see that middle cerebral artery supplies the majority of the frontal and parietal as well as the temporal lobe. Also, occipital lobe is supplied with exception of the small rim of tissue along the posterior margin of the hemisphere. So we could say that middle cerebral artery is largest vessel of the brain, and if circulation in this vessel is affected, it may result in damage of the extensive area of the cerebral cortex. Peripheral part of the hemisphere is supplied by the anterior cerebral artery follows the corpus callosum and gives off callosomarginal branches and pericallosal branch, and supplies medial aspect of the frontal lobe, medial aspect of the parietal lobe, and part of the sometimes part of the occipital, but in majority of cases, boundary of the middle of the anterior cerebral and posterior cerebral follows this deep sulcus. Posterior cerebral artery supplies the medial aspect of the occipital lobe and temporal lobe, and with extensions to the convexity, small extensions to the convexity of the hemisphere. Important is that posterior cerebral supplies the primary visual cortex, and this is calcarine artery, which lies in the deep of the fissura calcarina, and if this artery is closed or circulation decrease in this artery, follows damage of the primary visual cortex, which may result in partial or complete total blindness. Inferior aspect of the hemisphere shows that frontal lobe is mainly, basal aspect of the frontal lobe is mainly supplied by the middle cerebral, medial part by the anterior cerebral, and anterior pole of the temporal lobe is supplied by the posterior cerebral. Rest of the inferior surface of the temporal lobe is supplied by the posterior cerebral artery. Concerning to the venous drainage, arterial blood after passing through the arterioles and capillary net is conveyed from the brain via veins, and these veins generally opens to the sinuses. And you see that the veins from the convexity of the hemisphere opens to the superior sagittal sinus, and superior sagittal sinus, its continuation is to the transverse sinus and sigmoid sinus opens to the internal jugular vein. In addition to superior sagittal sinus, there is superficial middle cerebral vein, which receives smaller veins from the lower half of the frontal and frontal lobe, and after that, this superficial middle cerebral vein, its continuity is on the basal aspect of the hemisphere. Part of the cortical veins opens also to the superior anastomotic vein of Trollard, which interconnect the superficial middle cerebral and superior sagittal sinus, and part also opens to the inferior anastomotic vessel of Labin, which interconnect the Roland, which interconnect the Trollard vein, superficial middle vein, and transverse sinus. This aspect of the hemisphere shows that several veins draining the basal part of the frontal lobe opens to the cavernous sinus together with the sphenoparietal sinus and together with the superficial middle cerebral vein, and blood from the cavernous sinus is drained, which is situated at the lateral aspect of the body of the sphenoid bone. Blood from these cavernous sinus is distributed to the superior petrosal sinus and via inferior petrosal sinus to sigmoid sinus and finally to the internal jugular vein. Deep cerebral veins drain the basal ganglia, drain the thalamus, and also choroid plexus, and these veins opens to the straight sinus, and straight sinus opens to the confluence of the sinuses and to the consecutively blood is conveyed to the transverse and sigmoid sinuses. So blood from the deep of the brain is conveyed, is drained to the confluence of the sinuses. Whole brain, hemisphere, and cerebellum and spinal cord is surrounded by liquor cerebrospinalis, so cerebrospinal liquor. This fluid is produced in the choroid plexus and is contained in the ventricles and in the subarachnoid space. Together, there is circulating 140 milliliters of the liquor. Function of this fluid is physical support of the brain, because brain in fact floats within the fluid. It's not sitting on the base of the skull, but between the base of the skull and bones of the skull is layer of the liquor, layer of the fluid. In addition to this very important physical support of the brain, liquor represents the channel for chemical communication within the CNS. So substances from the neurons may be trained to the fluid and via walls of the ventricles back to the neurons. So two main localization of the fluid is subarachnoid space, which is between the pia mater and arachnoidea, and subarachnoid space is traversed by the septa, and out of the arachnoid is dura mater. So outer layer is dura mater. Internally to the dura mater is arachnoid membrane. After that follows the subarachnoid space, with circulating the liquor, and last layer is pia mater, which completely which contact the surface of the brain and adhere to the surface of the brain. In the spinal cord is from previous pictures evident that liquor circulates in the subarachnoid space. Subarachnoid space. In the spinal cord is subarachnoid space between the pia and arachnoidea, which adhere to the dura mater. So in the vertebral canal is epidural space. Following is dura mater, subarachnoidea, which adhere to the dura, follows the subarachnoid space, and last layer, last covering is pia mater. Important is that subarachnoid space, this one, which is in black color, projects along the roots and surrounds the origin of the spinal nerve together with the spinal ganglia. So, subarachnoid space project to the dural sheets, which are internal layering of the dural sheets is arachnoidea. So subarachnoid space extends to the dura sheets. Dura sheets are evident in examination of the spinal cord, the vertebral canal is using of the magnetic resonance. Yes, you see that is relatively short. There is extension of the dural sheets with accumulation of the liquor. There is very short dural sheets and all similarities here. This is this distribution size and generally diameters of the dural sheets are analyzed and inform us about some pathology which may compress the spinal cord roots, eventually origin of the spinal nerve. In addition to subarachnoid space, liquor is circulating in the ventricles, which are in deep of the brain. The ventricles are several. First one is lateral ventricle, which is right and left. Lateral ventricle is divided to the anterior horn, to the body of the lateral ventricle, to the trigonum of the lateral ventricle. And trigonum is an area from which originates the posterior horn of the lateral ventricle and the inferior or temporal horn of the lateral ventricle. From the lateral ventricle, fluid is distributed via interventricular foramen to the third ventricle, which is between the right and left thalamus, and third ventricle project by two recesses. Optic recess is anteriorly placed and infundibular recess is posteriorly and surrounds the infundibular region of the hypothalamus. This small protrusion indicates the position of the anterior commission. From the third ventricle, liquor is distributed to the to the Sylvian aqueduct, which opens to the fourth ventricle, and fourth ventricle projects with fastigium toward the cerebellum, and from the fourth ventricle, laterally and caudally project to recesses, lateral recesses, and continuation of the fourth ventricle is spinal cord canal, which passes through the whole length of the spinal cord. Production and resorption of the liquor must be in the equilibrium. So production is liquor is produced in the chorioid plexus, but is reabsorbed blood uh, and concretely to the sinuses. So to the sinuses opens protrusions which are named arachnoid villis, and via this villi is subarachnoid liquor fluid, subarachnoid fluid, reabsorbed back to the venous blood. Ventricular system and subarachnoid spaces of the brain are visible well on the magnetic resonance investigation, namely T2, T2, and because this this method shows liquor in the give liquor white signals. So white signals around the hemisphere in this cerebrospinal fluid. You see that cerebrospinal fluid enters also to the surface of the cingular gyrus, and these dark spots are branches of the middle cerebral artery. This one also, and these are branches of the anterior cerebral artery. So in addition to liquor, we could observe also vessels and some larger vessels, some larger branches of the vessels. In addition to subarachnoid space, liquor is situated in the ventricles. It is lateral ventricle, it is anterior horn of the lateral ventricle. This is posterior, this is pars centralis, central part of the lateral ventricle, which is bounded by the corpus callosum, nucleus caudatus, and dorsal aspect of the thalamus. Green color indicates the presence of the liquor. See that liquor is in the third ventricle, and from the third ventricle is distributed to the fourth ventricle. It is a picture showing the central portion of the lateral ventricle. This caudate nucleus thalamus, it is interventricular foramen and communication between the lateral ventricle and third ventricle. This small bridge is interthalamic adhesions. It is gray matter which bridge the third ventricle, and you see the vessels on the surface of the insular cortex, and this part of the temporal lobe is evident temporal horn of the lateral ventricle, and to the lateral horn project the hypocampal formation. On this other side, it is evident the same. This is a schematic picture showing boundary over the cornu temporal horn. So button is formed by the hypocampal formation, and upper surface is formed by the hemisphere. Lateral aspect of the lateral ventricle. This is liquor around the cerebellum and tectum, and in some areas is this is some areas suprarachnoid space exhibit dilatation or accumulation of the liquor, and these areas are names cisternae of the subarachnoid space. This is cisterna around the tectum of the midbrain, cerebellar, cisterna between the oblongata and cerebellum, and these are cisternae situated at the basal aspect of the brain. So, with this picture, we are terminating this